Good afternoon, everyone. My name is Pascal Laurent, and I am a commercial marketing manager at Oxford Nanopore Technologies. It gives me a great pleasure to welcome you all to our Core Lab webinar series dedicated to core facilities and service providers. Here, we will be exploring the latest sequencing application developed by Oxford Nanopore Technologies and how they can help your sequencing laboratory address the key demands of the research community. With the support of Oxford Nanopore Technology, you will be able to adopt and implement application quickly, streamline your workflow, and accelerate every stage of the sequencing process. Oxford Nanopore is here to help. We want to help core labs provide better answers to researchers' scientific questions, offer cost-effective solutions and faster results, be a valued expert, and an essential resource for your research community. Oxford Nanopore is evolving to build effective partnership with your core lab. The Core Lab program supports the growth of your facility and is a partnership to maintain your genomic technology expert status. We want to help you attract new research projects to your core lab, expand your application menu and offering, accelerate your project and time to completion, and maintain your expert status with the sequencing technology. The aim of this webinar series is to help your core business expand its technical knowledge, provide guidance for sequencing projects, and give you a fresh opportunity to engage with the research community. Our 2024 Core Lab webinar series comes in 3 parts. Today, we will start with the comprehensive analysis of the human genome before moving into cancer application and accuracy of nanopore sequencing. Today is our first Core Lab webinar in this series, Comprehensive Analysis of the Human Variants and Cancer Genomes with Multi-omics Nanopore Sequencing. I'm thrilled to be joined today by Dr. Mikhail Kolmogorov from the National Cancer Institute, part of the National Institute of Health in Bethesda, and my colleague Etienne Raimondeau, Technical Product Manager at Oxford Nanopore Technologies. After the talk, Michael and Etienne will be taking part in a live Q&A session, so please add your question into the Q&A box, which you will find at the bottom of your screen. You can also upvote the question you would most like answered, and we will try to get through as many as we can. This webinar is being recorded and will be made available on demand shortly after the broadcast. So, to start off today, I would like to introduce Dr. Mikhail Kolmogorov. Before joining the Cancer Data Science Laboratory in January 2022, Mikhail was a postdoctoral fellow at the University of California, Santa Cruz, supervised by Dr. Benedict Patton. Mikhail completed his PhD in September 2019 in computer science from UC San Diego under the mentorship of Dr. Pavel Pevsner. His talk today is NAPU and Severus, Accurate and Complete Germline and Somatic Structural Variation Coding.
Thank you, Pascal, for the kind introduction. It is my great pleasure to be here, and thanks a lot for inviting me to present this work. And in this talk today, I will be talking about structural variant analysis in 2 different contexts. I will be talking about germline structural variation and how can we analyze it using the NAPU pipeline. And in the second part of my talk, I will focus about analysis of somatic structural variations with Severus. So this is the first part, germline structural variation. And this has been developed as a part of the CART initiative at NIH, which stands for Center for Alzheimer and Related Dementias. This is a new center. And as you can probably tell from the name, we are broadly interested in structural variants that contribute to various neurodegenerative disorders, and we want to use long reads to study these structural variants. The ambitious goal is to sequence over 4,000 brain samples from various biobanks, various cohorts, and there are 2 obvious problems. First, we need really scalable long-read sequencing protocols And secondly, we need efficient algorithms to analyze this data. And so this work that I will be presenting has recently been published. So if you want to learn more details, I encourage you to check out this paper that you can see on the left. And I want to acknowledge the entire CAR team. This is a huge team effort. And in particular, Cornelis and Kim, who are leading the long-read sequencing initiative at CART and really responsible for producing this amazing data and developing protocols. And speaking about protocols, the goal was to use a single flow cell per sample so that we can really scale it up. And we have a little bit of trade-off between the yield and the sequence length. So what we settled was the yield at around 30 to 40 x. Let's translate to about 100 to 120 gigs of data, and the real length and 50 about 30 kb. So this is certainly not ultra long, but as you can see, this is actually very much sufficient for the analysis of small structure variants in the most parts of the genome, perhaps excluding the longest segmented applications and centromeres. So these are the numbers for R9. For R10, we have somewhat similar numbers. It required some tweaks, but I think we are, with the recent updates, we can actually push the yield with R10 up. So here, this is the protocol, and this is how we analyze this data. This is the Napo pipeline. This pipeline has been written in Vidal, and it runs on Terra in the cloud, but it's also really easy to run it on a computational HPC server. So very briefly, it consists from the mapping-based part and assembly part. So mapping-based, we start from mapping reads using Minimap. We call small variants using Pepper margin invariant. And in R10, we just using the variant. We also can call the haplotype-specific methylation from this phased alignment. And for the assembly-based, we're using a combination of Shasta and Habdup, a new tool that we have developed. And here are some details how it works. In fact, we can start with any haploid assembly, so we can use either Shasta or Fly. We use Shasta in the CARD initiative because it runs faster. We map original reads back to the assembly. We call SNPs. We phase them, and we haplotype the reads. So we split them into two bins based on their haplotype. And then each set of reads is used to locally reassemble the initial haploid genome. This gives us really a deployed assembly, which is locally phased. So this is not chromosome scale phasing, but is the phase block length is about 1 to 2 megabases. So this is pretty good and does not require any additional data. And then we call structural variants using these assemblies, using another tool that we have developed called Habdiv, but it's really similar to some other tools that are out there. And so finally, we merge small and structural variant calls together and phase them in a harmonized VCF using this step. And the final output, you get small variant calls, structural variant calls, methylation calls, and deployed de novo assembly. So we of course wanted to measure how good these assemblies are, and this is the structural variant benchmark. Structural variant calls that were produced using these assemblies, and we benchmark it against the genome in the bottle call set. As you can see, the benchmarks on the right, our Habdup scores were slightly improved over the mapping-based approach, were slightly below the HPRC-based assemblies produced by a mix of different technologies. But as you can see, the consistency is overall pretty high, and we're pretty happy. Just for the sake of comparison, this is how the best Illumina-based structural variant callers are performing. We know that there are limited on germline structural variants, and I will actually come back to this when we'll be discussing the somatic structural variant calls. And of course, using these assemblies, you're not just getting single structure variants, but it allows you to analyze more complex loci with clusters of different small structural variants. And this is just one example showing our nanopore-based reconstruction on the top track and the HPRC-based reconstruction on the bottom track. As you see, it's a fairly complex locus, but the assemblies look really identical. And of course, since we have a lot of assemblies, we are trying to construct pangenome graphs. So here are two examples of highly polymorphic MHC locus and also IGH locus. IGH is interesting in this case because most of the current large-scale assembly project, they use data that is derived from the blood-derived cell lines. So they're derived from B cells. And B cells actually have a rearranged IGH locus, making it difficult to analyze the germline evolution. But since we have brain tissues, it's actually in a germline state. So this gives us a very nice resource for the groups who are interested in studying this using immunogenomics approaches. So as a summary for the NAPU pipeline, we aim to develop the really scalable sequencing protocol and combine it with a computational pipeline. It works well for both R9 and R10. It can generate de novo deployed assemblies with an asterisk that they are not very comprehensive in the most repetitive regions, such as centromeres and long segmented applications. And for that, we perhaps need ultra-long reads. But here we make this trade-off between the yield and read length to really make it scalable. Using these assemblies, you can detect structural variants with very high F1 scores, high consistency. You can get SNP calls comparable to Illumina. You can get haplotype-specific methylation. The remaining challenge is, of course, short indels. It has improved quite a bit with R10, and it looks like there's more exciting updates that are coming. So we think that pretty soon we will have very nice and comprehensive indel calls as well. So now we'll move to the second part of the talk, and I will be talking about somatic structural variations analysis in cancer genomes. And I will talk about the tool called Severus that we have developed. So we know that somatic structural variations is an important contributor to cancerogenesis, and in fact, about half of the driver events actually overlap with structural variants. And in the cancer, in the context of somatic variation, in the context of cancer, the patterns and the structures of this variation and mutational process can get much more tricky. As you can see on the right, we have quote-unquote simple variation that we typically observe in germline, such as deletions, insertions, duplications, inversions. In cancer, we often observe more complicated patterns, driven by different mutational processes, such as chromoplexy, when there's a balanced exchange of DNA material between multiple chromosomes, or chromotripsis, when the chromosome is shattered and then reassembled in a different order. So we are, of course, interested in studying this, interested in complicated processes with long reads. And I wanted to start with an example of one particular mutational process called breakage fusion bridge. And it is prevalent in different types of cancer, in particular cervical cancer, but in other types as well. And it has been known for many years, and of course, in fact, originally it was described in maize by a long time ago. So the process starts from a telomere loss, followed by a fusion of sister chromatids because the chromosome becomes because there are double-stranded breaks. This forms a bridge, and once during the cell division, the cell tries to pull this apart using the centromeres, eventually creating a break. But this break happens not in the exact location of the bridge, creating a duplication. And this also results in a fallback inversion. On the right, you can see a typical coverage profile. And in fact, this has been extensively studied in the concept of cancer from the coverage-based analysis using Illumina reads. But we think with long reads, we can actually approach this slightly differently. So here's an example of long read alignment, which is phased. We see one haplotype is completely untouched. The other haplotype has a fallback inversion and a loss of heterozygosity on the telomeric side, just as we expect. So because after this break, the telomeric portion of the chromosome is lost. And we can see clear breakpoints of this inversion. It really enables us to go to have a sequence level resolution and study in detail what is happening around these breakpoints. And what we found was that there actually sometimes templated insertions around these breakpoints. And this kind of makes sense because you have double-stranded breaks and you have all kinds of mutational processes going on, and maybe you can get a templated insertion from a template sequence from another chromosome. So we studied this in a panel of 9 cervical cancer cell lines. And this is a collaboration with Michael Dean lab. As you can see, this has recently been published. So I encourage you to check out this paper for any details. In total, we characterized 13 such amplifications. 4 of them have these templated insertions that were otherwise would be very difficult to see using short reads. We also found that this mechanism in particular seems to be responsible for amplifying YAP1 gene, which is linked to a 10-year earlier age of diagnosis of cervical cancer. And we know this because we went back to the TCGA coverage profiles, and the coverage profiles look very consistent with the BFB origin of this amplification. We see a stair-like jump in coverage, and then we see drop of the telomeric side of the chromosome, supposedly corresponding to the loss of heterozygosity. So once we know what to look for, it's really clear once we go back to the TCGA coverage profiles. So how did we detect all these rearrangements? So for that, we developed a new tool called Severus, which is a somatic structure variant caller for long reads. So it's really developed with the complex somatic variants in mind, and that's differentiates it from other popular tools such as Nifls and QTSV. Severus by design supports tumor normal analysis or multiple tumor sample analysis. It produces haplotype resolved calls, and it enables classification of complex structure variants. Severus is available and on GitHub using this link. So if you're interested, I encourage you to take a look. I will briefly describe the algorithm. And we start from really detecting the germline variants in the normal sample, if the normal sample is available. If not, they actually can be detected from tumor sample as well. And we use this variant to haplotag the normal reads. And then we extend this phasing and haplotag into the tumor sample. Here you can see on the top is, phase normal sample. and on the bottom, tumor sample with one haplotype is completely untouched. The other haplotype has an inversion. And then using this, the breakpoints that we can detect, we build something that is called a phased breakpoint graph. This type of graphs has been extensively used to compare different genomes or structure variants within the same genome. But haplotagging and a long grid-based phasing enables us to simplify this graph even further using the phasing information. So very briefly, as you can see a schematic representation of preference on the right, you can think about these blocks as maybe genes. And we next extract the alignment information from reads. And so some reads will align in full, some reads will have split alignments, multiple alignments. And the split alignments basically will define additional edges on this graph, as you can see here in green. And we basically built a paternal and maternal copy of this local breakpoint graphs. And then by analyzing the clustering and analyzing the structure of this graph, we can detect and classify the structural variants. So why is the graph approach so useful? And I actually will give you a clinical example. So this is a collaboration with Mithat Faruqi at Children's Mercy Hospital. They are using a long read sequencing as a pilot project for diagnosis of pediatric leukemias. And this is an example of initial undiagnosed acute myeloid leukemia case. And by undiagnosed, I mean it was subject to standard genomic panels, such as microarray, karyotyping, FISH, small variant calls from Illumina whole genome. And this all came back negative. This did not reveal any apparent drivers. They have it sequenced with long reads, and we ran Severus. And Severus detected this highly rearranged locus between the, well, actual two, between chromosomes 10 and 11 with 5 rearrangements. And in fact, one of these rearrangements was a diagnostic signature. The fusion between KMT2A and ML1010, it classifies this AML into a clinical subtype and is important for diagnosis. And it is interesting that, in fact, specialized FISH probes did not return a positive signal here. And we think that this is because the structure of this locus is so cryptic that standardized probes just did not work. But it's very easy to see from this graph-based representation using Severus. And to evaluate this in a systematic manner, we sequenced a panel of cell lines. You can see 5 cell lines here. We in fact have one more that is in progress right now. We sequenced it with 3 different technologies: Illumina, PacBio, and Nanopore. And we ran various tools. We ran the state-of-the-art Illumina-based tools. We ran Sniffles, and we ran Savannah and Nanonomas V, which are recently developed somatic structure variant callers for long reads. Well, first of all, you can see that Severus is, in fact, consistently either outperforming or performing similar to other tools in both Nanopore and Hi-Fi categories. And most of the time, the long read-based approach is outperforming Illumina. But Illumina is actually, on many samples, is pretty close, which tells us that the patterns of somatic variations in cancer are quite different from the germinal structure variants that we have discussed in the first part of this talk. And just to note, these cell lines initially did not have any ground truth benchmark. So what we did, we took a consensus of multiple tools and technologies to arrive to a set of confident calls. And we benchmarked all the tools against this set. So we looked a little bit deeper into the patterns of errors that different technologies and different tools are making, and which are summarized here on this figure. So I will walk you through. Every dot corresponds to the F1 score for different cell lines and different tools. And the structural variance was stratified based on the challenging patterns, different challenging patterns. For example, as you can see for insertions, Illumina methods are not doing as good as long-read-based methods, and it's not perhaps not so surprising. Interestingly, homologous repeats, so when there is homology between the break-in junctions for structural variants, might be more challenging for Illumina-based methods. In the repetitive parts of the genome, such as segmental duplications and tandem repeats, primarily VNTRs, long reads are doing a bit better, but as you can see, there is quite a bit of disagreement within the long reads methods as well. Of course, the variants with low allelic frequencies are challenging. Short structural variants, so structural variants between 50 and 100 base pairs, are somewhat difficult. And an interesting category is the chains of rearrangements or chains of structural variant junctions, when you basically have structural variants that jump from one chromosome to another and another, basically chains of these rearrangements. And this was challenging to detect world tools, a little bit more challenging for Illumina. And finally, on the bottom right, you can see the variants that have no apparent complications, and most tools indeed performed very well on those variants. And in the end of my talk, I will wanted to briefly highlight another method that is in development called Vahan. It's already available on GitHub. So Vahan is really aimed for the chromosome scale copy number analysis using long reads. And because we have phasing information from long reads, we can generate haplotype-specific copy number profiles. As you can see on this slide, this is an example from one of our cell lines. So with that, I'd like to acknowledge a lot of people who contributed to this work that I've been presenting. Of course, my group, we are a very young group. We started really a couple of years ago, and I was very fortunate to recruit these amazing fellows. And we also have a lot of collaborators from different institutes. And with that, I'd like to thank you for your attention, and I will be happy to take any questions.
Thank you, Michael, for your wonderful presentation. It's now time for our second speaker today, Etienne Raimondeau, Technical Product Manager at Oxford Nanopore Technologies. His talk title today is Accelerating Clinical Research with Streamlined End-to-End Protocol for Nanopore Sequencing. Over to you, Etienne.
So welcome to this presentation about end-to-end protocol for nanopore sequencing, enabling to accelerate clinical research for human genetics. So first, I want to bring everyone to the same speed and explain briefly our technology work. So nanopore sequencing is a label-free single molecule method of sequencing native DNA or RNA. So the method happens in real time, and you can have high throughput sequencing. And so the way this works is that you have a motor enzyme here in purple that is bound to the DNA or RNA, and it will unwind the DNA or RNA through the pore. And there's an electric signal applied on the flow cell, and as the DNA moves through the pore, the electric signal is disrupted. And this disruption in signal is then turned into ATGC, into DNA bases. And this process is called base calling. And so because we just measure an electric signal, there's no laser, no optics. This method is basically happening in real time. So as the DNA go through the pore, you can detect the sequence and base call it. So we have different devices enabling nanopore sequencing. So different flow cell size depending the type of experiment you want to do, from small output to larger output. For example, required for human genome, so from fungal to PromethION Flow Cell. And similarly, we have different type of devices depending the scale of your project, from a MinION device all the way to a PromethION device, which enable high throughput type of studies. So the high accuracy of Nanopore enable different type of investigation. I just want to remind everyone about the accuracy number that the platform is achieving currently. And so in terms of single row read accuracy, so a single strand of DNA going through the pore, we're achieving Q24. And once this is paired with the complementary strand or the follow-on strand for what we call duplex, this is boosted to Q31. And so this enables us to have a very accurate consensus accuracy. So for example, for telomere-to-telomere assembly, we're at Q47. And we have great F1 score for human variations. So I put the number here for SNP detection, indel, and structural variant. Something I didn't mention in the previous slide is that we're also great at sequencing methylation. So the methylation is embedded in the nanopore signal, so there's no additional prep that you need to do to be able to obtain this the methylation signal. You just need to check a tick box on the software. And the accuracy for 5MC is at the moment 99.7. So I put the QR code at the bottom of the slide where you can go on our website and explore this number further. So in terms of human genomics, we have different kind of investigation that we can enable, from targeted sequencing if you're only interested in few genes or few section of a gene, all the way to telomere assembly, where you can basically assemble a genome. And what we're talking about today is standard whole genome sequencing at about 30x coverage, and so that's enough to reliably capture SNP, indel, and methylation. So the way the end-to-end protocol works is, of course, starting from starting material. So in this case, that would be cells or blood. Then you go through, of course, sample extraction, which is optimized to extract high-quality long DNA. You go through a site selection step, and then this DNA is going through a library preparation where we add the sequencing adapter with the motor that unwind the DNA by ligation. So that's a fairly short step that takes about 60 minutes. And then the sample is sequenced on a device. So in this case, that's a PromethION Flow Cell. We recommend to sequence for 72 hours. But again, the data is generated in real time, so you can interact with your data while it's being generated. And then we recommend to use epitomy for analysis. So we have a workflow called human variation, which can analyze your FASTQ files. So at the bottom of the slide, I added the links to the full-length protocol. So we have two types of protocol, one for 10 KB fragment lengths, and one for 30 KB, and that's the one I'm focusing more on today. We also have some additional resources on our resources center on our website, and I put the link on this slide as well. So in terms of extraction and library prep, we recommend to use QIAGEN PureGene kit, which in our hands generated the best high molecular weight DNA for this workflow. The next step is a size selection. So in order to retain the 30 kb strand and above, we recommend using the short fragment eliminator, or SFE, which can be found on our store. So we estimate the sample extraction and size selection take about 300 minutes. And then you can move to the library prep. So as I mentioned, that's attaching the sequencing adapter. This is done by ligation using the ligation sequencing kit, or LSK114. So that's a PCR-free method that is optimized for output. And if a user wants to maximize the output of their flow cell, they can do so by washing and reloading library every 24 hours. So we found in the lab that it takes about 3 hours for a single researcher to QC and manually prepare 24 samples and put them on the flow cell, but this can be automated down to less than 3 hours. With very little hands-on time. And so with this protocol, using PromethION Flow Cell, we recommend to sequence until you achieve 30x depth of coverage. And so this is done using a device called PromethION. So we have different scale for this device, depending on the scale of your project. The workflow is working for all of them. If you require very, let's say 200 genome per year, then a device called the P2 is completely sufficient. Now, if you have larger scale project to sequence 2,500 sample per year, then we would recommend a P24. And if you have more than these, we then recommend a P48 device. The price per flow cell is competitive. So at the moment it's $690 per genome, and of course this price would come down in the future once we can do 2 genome per flow cell. So in terms of analysis, we recommend to use Epitomy. So this is a very intuitive software. So we have a graphic interface, but you can also run it via command line. And so this can be run while the sequencing is still ongoing in Mino, or you can do it after the run. So the way it works is that you point out your FASTQ file to the software, and it will basically output nice HTML report depending the type of analysis you choose. So we can do SNVs, SV, ST, as well as copy number variation. If you want to do methylation, you will have to select, make sure that you select methylation sequencing during your sequencing in Mino. And we can also do phasing. And so on top of the HTML report, you also get BAM files, and these files can be visualized in third-party software where you can carry on further analysis on the sample. And so that's it for this end-to-end workflow, and thank you for your attention.
So thank you again, Mikhail and Etienne, for joining us today. If you want to learn more about today's topic, you can find more information at nanoportech.com/application/technique/whole-genome sequencing. Again, today, Mikhail presented the analysis of germline and somatic structural variation, and Etienne described the end-to-end workflow that are used to detect germline structural variants. The next webinar in the Core Lab series will be looking at cancer application, where we will be exploring somatic structural variation further. I do hope you can join us. Detail of these broadcasts will be announced shortly. That brings us to the end of today's Core Lab webinar, and I do hope you have found it useful. This webinar, along with all of our previous broadcasts, are available for you to watch on demand. So please visit Oxford Nanopore website to check them out. You can also find more information about our other upcoming webinars and knowledge exchange on the Nanopore website. If you have further questions about your project or getting started with nanopore sequencing, please contact support@nanoporetech.com. We would really appreciate your feedback on this webinar. Please head below to the poll tabs next to the Q&A tab, where you will find a quick survey on how you found today's broadcast. This will remain open once the webinar ends. That was it. Thank you and enjoy the rest of your day.